Indonesia, an archipelago rich in biodiversity, is home to a vast array of plant species. The tropical climate and diverse ecosystems, ranging from rainforests to coastal regions, make Indonesia a significant botanical hotspot. The unique flora contributes to global biodiversity and plays a vital role in the daily lives of its people, providing food, medicine, shelter, and supporting cultural practices. The rainforests harbor many endemic species, making botanical research in Indonesia crucial for understanding and preserving this natural wealth. Chlorophyll is a green pigment found in the chloroplasts in plant cells, playing a crucial role in the process of photosynthesis. Through photosynthesis, chlorophyll converts light energy into chemical energy, which is essential for plant survival and serves as a primary source of nutrition for animals and humans. Photosynthesis produces oxygen and glucose, supporting life on Earth. However, leaves often undergo dehydration due to environmental factors such as heat and drought. Dehydration can lead to significant changes in cellular structure and the degradation of pigments, including chlorophyll.
Chlorophyll can be separated and identified using simple paper chromatography techniques. Chromatography is a vital tool for chemists and many other scientists who need to separate mixtures and identify their components. The ability to qualitatively identify the substances found in a sample can be critical. For instance, an environmental chemist investigating samples of polluted groundwater will need to know which toxic ions might be present. Chromatography is often one of the first techniques used in such situations. This method allows the separation of various types of mixtures into their pure component substances. By comparing the results to a standard sample, each component can be tentatively identified.
So the main question of this study is, how does dehydration affect chlorophyll retention in leaf tissues? And the objective of this study is to analyze the impact of dehydration on chlorophyll retention in leaf tissues. This study aims to understand how dehydration affects the presence and retention of chlorophyll in leaves, which is important for various applications in agriculture, food technology, and environmental research. So how do we know the presence of chlorophyll in leaves? The method that will be used is a simple thin layer chromatography method. The following are the tools and materials that will be used and the steps. Tools and materials include alcohol 70%, leaf sample, tissue, cutting board, glass, mortar and pestle, filter, receptacle, paper, sticks, knife, and ruler. The first step is to take a leaf sample and wash it using water. Next, some leaves are sun-dried until they become wilted and slightly dry. 3 types of samples were obtained. The 3 samples were fresh, wilted and dry leaves. Leaf samples were cut into smaller pieces to ease the grinding process. 3 samples of ground leaves were collected. Pour the sample onto a filter lined with tissue. Then dissolve the leaf sample using alcohol. Repeat the same steps for the other leaf samples. 3 leaf sample extracts were obtained. The next step, insert the paper that will be used as chromatography media. Wait until the color movement occurs on the chromatography paper. After a while, the color movement begins to occur. After the color movement is complete, remove the chromatography paper from the glass. Chromatography results were obtained on leaf samples.
Based on the results obtained, the RF value can be calculated. RF value is a ratio that represents the relative distance traveled by the point compared to the distance it could have traveled if the point had moved in the direction of the solvent. The RF value for each dye is then worked out using the formula. RF is equal to the distance traveled by solute divided by the distance traveled by solvent. For example, if the red component traveled 1.7 centimeters from the baseline, while the solvent had traveled 5.0 centimeters, then the RF value for the red dye is RF is equal to 1.7 centimeters divided by 5.0 centimeters, resulting in an RF value of 0.34. With the same calculation, the RF value of the samples is obtained as follows. For fresh leaf sample, the following values were obtained. For the total solvent distance of 5.5 centimeters, the RF value for carotene was 1.00, xanthophyll 0.91, chlorophyll A 0.36, and chlorophyll B 0.20. For wilted leaf sample, the following values were obtained. For the total solvent distance of 5 centimeters, the RF value for carotene was 1.00, xanthophyll 0.82, chlorophyll A 0.26, and chlorophyll B 0.08. For dried leaf sample, the following values were obtained. For the total solvent distance of 5 centimeters, the RF value for carotene was 1.00, xanthophyll 0.80, chlorophyll A 0.16, and chlorophyll B 0.06. The data obtained is presented in the following diagram. It can be seen that the RF value of chlorophyll decreases as the leaf tissue is damaged due drying. In fresh leaves, we observe distinct bands of chlorophyll A, blue-green, chlorophyll B, yellow-green, and carotenoids, orange. In wilted leaves, the intensity of chlorophyll bands significantly decreased, indicating a reduction in chlorophyll content due to dehydration. In dried leaves, there was a significant decrease in the intensity of the chlorophyll bands, indicating further degradation due to more extensive dehydration. However, carotenoids were relatively stable, maintaining their intensity even under stress condition. The results show that dehydration leads to a degradation of chlorophyll, highlighting its sensitivity to environmental stress.